Hi, this is Alicia Armenta for Bio 225. It is now, oh my gosh, what's the date? 3/25/2018. This is for lab number 4, microscopy, tongue twister, for microbiology, use and function. And I just completed the lab last night, and this is my video for showing you the slide images that I took. The lab took me 6 hours instead of 3. I'll get to that in a minute. So I didn't record during that because the process just took too long and the video would have been too long. And I also didn't want to give like several snippets of the video because it didn't require incubation and all that. So I wanted to make it easier for you. So I'm going to be showing you the images on my Google Drive that I uploaded through my cell phone, and I will explain what we're viewing. So here is my Google Drive. You can see my pictures that I took from my previous experiment here. So we're going to start with this one, the letter E. This is for exercise 1. And this is on the scanning power, and as you can see, the square is visible in this power and all of the E. You can't really see the details of the E yet, or the edges. You can just tell that it's an upside down E. And then here we have the low power objective lens. And now the square has disappeared and all you can see is the E. The edges are becoming a little more refined. You can make out more of the composition of the E. And then we have high power. You can't see the majority of the E. This is just like the middle of the E that goes like that. So that's in there. It's fuzzy on the edges now. You can tell they're not as sharp as they appeared to be. Now this is using the oil immersion lens. This is a total magnification of 1500, and I described it as looking like a fuzzy caterpillar because it actually, you can see like the level of ink from thick to thin. So I thought that was pretty neat. And then this is for the amoeba slide. This is the low power. And then this is the, excuse me. This is for the scanning power, as you can clearly see. This is the low power. And you can see that there's a smaller number of cells. And this is high power. I chose this one to include in my lab report because you can see the entire cell and its structure, which is important. Although you could use this one, which is the oil immersion, and it gives more specificity in the internal structure of it, but if you want to see the entire thing, you would go with high power. So this is the penicillin with candida. This is scanning, and you can't really see the flakes that we want to take a picture of, so we're not going to use that one. This is the low power. So the specimen's becoming more visible. And then this is high power, getting a little bit clearer here, but we still want it to get a little closer, so I used the oil immersion lens, which is this image you see here. It's very clear, you can see the structure. It's beautiful, my best picture yet in this lab. Okay, so then here, this one was a little difficult because of the flare. This one is the yeast, and you can see the individual cells, tons of them. It's becoming more refined as you increase the power So the previous image you saw was scanning. This is now low power. This is high power, a little more refined. And this is my favorite image because of all the blues. This is the oil immersion lens. I chose this image because it showed the entirety of the cell as well as the internal structures. This is the Bacteriosporillum. This is the middle of the slide on the scanning power, excuse me. This is on low power. You can't really see a lot of the detail yet, so we're just going to keep increasing the power and focus. So this is high power, and we want to see a little more detail, so we're going to increase it one more time. And when we go to the oil immersion lens, this is the image I used with a total magnification of, again, 1500, and this is what we want to see. So I used that image. Okay, so this is the Bacteriococcus slide. I described my issues with it before in my lab report. I went into more detail. It took me 3 hours just to get this specimen. This is scanning power. I did not see a single thing on this slide. I had no trouble at all with the other slides locating the specimen. So I'm thinking either there was an error and this one just wasn't included with the specimen, or that's it. I couldn't really tell. So this was after 3 hours. This is a low power image. I did not use that, of course. This is high power. This I did use because it shows the entirety of the structure and the internal portions of it. But just to show you, this is the oil immersion lens. So, I mean, this is what I got. Hopefully it's something. Okay, so this is the Bacteriococcus. I'm having a brain fart here. I don't know if I just talked about the bacillus. The last one I just talked about was bacillus. This is supposed to be coccus. And this is the scanning power. Becoming larger now. This is the low power. We still want to see it larger. This is the high power. Again, well, this slide, the only thing that had a small specimen or with color was this. So this didn't have a lot of specimen on it either. I was quite disappointed. And this is the oil immersion lens. I used this one in my lab report because it's clearer and bigger. And then this is my cheek cell on scanning power. It's very tiny, but it's there. That was the best lighting I could do. And then here, there's two of them now. They're getting bigger. That was the low power. This is the high power. We didn't use the immersion lens on this particular one because it wasn't required. So this is the power I used, the high power for my lab report, because you can clearly see the structure, all of it, and the internal portions of it. And then here we have the scanning view of my dental plaque cells. There's a lot of them. I got a really good sample on that one. Scrub the crap out of my teeth. Then we have the low power, and they're getting a little bit clearer, but we still want to see it a little bigger and closer up. So I used the high power here in my lab report because you can clearly see the structure and the internal portions of it, even though the cheek cells and the dental plaque cells were transparent. I made it a little difficult to actually see the cell, but I conquered and found it. So this was my entire lab. And thank you for watching.